Picture the Himalayas the way most people already do: snow, silence, and a handful of villages clinging to slopes where the air itself seems to be running out. You have probably seen the footage. A Sherpa guide, loaded down with gear, walking past exhausted trekkers gasping for breath at Everest Base Camp, 5,364 meters up. The air there holds roughly half the oxygen your lungs expect at sea level. You would be dizzy within minutes, nauseated, maybe worse. Your body would be screaming for oxygen it cannot get. He is not even breathing hard. Not him. The story we tell ourselves about that is simple. These are mountain people. They have always been mountain people. Same blood, same peaks, unbroken, for as long as anyone can remember, stretching back before recorded history even started counting. It feels less like a historical claim and more like a fact of nature, something as fixed and permanent as the mountains themselves. No migration, no mixing, just one people in one place since the beginning. Generation after generation, the assumption goes. That assumption sounds solid. It has never actually been tested. Here is the crack in that picture. Living in a place and descending from the people who first lived there are not the same thing. Nepal is not one population. It is dozens, layered from the lowland plains to the highest passes on Earth. So the real question is not whether mountain people exist. It is narrower than that, and much harder to answer. Are today's Sherpas the direct descendants of the humans buried in these mountains 1,000, 2,000, 3,000 years ago? Or did a different population arrive later, take up the same peaks, and simply inherit the name? A real possibility, and a genuine one. Here is how researchers actually test something like that. Not with legend, not with language, but with bodies. High in Nepal's Mustang region, in cliffside caves with names like Chokapani, Mebrok, and Samzong, archaeologists have recovered human remains dating across 3 separate periods, the oldest around 3,150 years old, the youngest closer to 1,250. In 2016, a team from UC Merced, the University of Oklahoma, the University of Chicago, and Uppsala University sequenced the nuclear and mitochondrial genomes of 8 of these individuals, publishing their results in the Proceedings of the National Academy of Sciences. In 2022, a second team, writing in Nature Communications, expanded that dataset to 33 ancient genomes, 41 ancient individuals in total, once both studies are combined. The method itself is straightforward. Take the genetic markers preserved in ancient bone, compare them against reference panels built from living populations across Asia. Whichever modern group the ancient genome statistically clusters with, that is the closest living match. It sounds simple. It is anything but. This should have been done decades earlier. It was not, for one blunt reason. Before 2016, there was no ancient DNA study of the Himalayan region at all. Not one. 0 prior data. A blank page. Ancient DNA breaks down fast in heat and humidity, and most of South Asia offers plenty of both. The Himalayas are the exception. The cold worked in the scientists' favor for once. Cold, dry, high-altitude caves preserve genetic material the way a freezer preserves meat. That is why Nepal is one of the only places on this entire landmass where DNA this old survives at all. Before elimination, widen the field. The comparison did not start with 2 options. It started with many, drawn from every corner of the region. There are the Nuar, the historic civilization of the Kathmandu Valley, with their own language, their own script, over 2,000 years of continuous city life. Nuwari is not closely related to Nepali at all, a linguistic island in the middle of the country. There are the Tamang, spread across Nepal's mid-hills, whose oral histories claim descent from Tibetan cavalry sent south centuries ago. Soldiers, not settlers, or so the story goes. Their name is even thought to derive from a Tibetan word for horseman. There are the lowland communities of the Terai, near the Indian border, living at barely 100 meters above sea level, closer in climate and culture to the Ganges Plain than to any glacier. Same country, wildly different genome, separated from the peaks by less than 100 kilometers. There are Han Chinese populations from the lowlands further east, sharing the same vast landmass that eventually rises into the Tibetan Plateau, same continent, same general direction the ancestors of every high-altitude population in Asia are believed to have come from. There are the Tibetans themselves, from the plateau proper, the group most people would guess first. If this were a betting game, most viewers would put their money here. And there are the Sherpa, concentrated around the Khumbu region near Everest, who by some estimates only moved into Nepal from Tibet around 500 years ago. 500 years. Recent, by the standards of this story. 6 candidate populations. 6 different histories, languages, and migration stories, all layered across one country. Genetically, these are not interchangeable. Some are separated by mountain ranges, some are separated by deep time, and somewhere in that list sits the population that will actually match the bones in the caves. Nobody watching this yet knows which one. Every one of them a real possibility. Given everything laid out so far, there is an obvious prediction. Nepal is a gradient, not a single block. Lowland to highland, plains to peaks, trade routes running through the middle of it for centuries. So the expectation is a blend. A mixed genetic signature reflecting that entire gradient, pulling a little from the Terai, a little from the trade-connected valleys, a little from the high passes, or, at minimum, a pull toward whichever group had the most historical contact with these specific burial sites, geographically and economically. That is how history usually works. Neighbors mix. Trade routes blur boundaries. Isolation is rare, and total isolation is rarer still. If genetics followed geography and commerce the way it usually does, these ancient genomes should look like a patchwork, not a single clean match. That is what should have happened. The elimination started with the group farthest away, and it did not take long. The Terai populations fell first. Genetically, they sit closer to the farming communities of the Ganges Plain than to anyone 60 miles north and 4,000 meters up. Same country, same passport, almost no shared ancestry. Distance on a map and distance in a genome turned out to be 2 very different measurements. Next came the Han Chinese comparison, and this one surprised even the researchers. You would expect some connection, given that Tibet and its plateau sit on the same enormous landmass. Instead, the ancient Himalayan genomes and lowland Han genomes trace back to populations that split apart somewhere between 44,000 and 58,000 years ago. 44,000 years is older than cave art in Europe, ruled out, and ruled out decisively. The Nuar came next, and this elimination stung a little, because the Kathmandu Valley has been continuously inhabited for over 2,000 years. Real history, real depth, a genuine civilization with its own script and its own language, unrelated to Nepali. But 2,000 years of urban life also means 2,000 years of trade, marriage, and mixing with lowland and valley populations passing through. The ancient mountain genomes did not carry that signature. Close geographically, wrong genetically. The Tamang were harder to dismiss. Their own oral tradition claims descent from Tibetan soldiers, cavalry sent south generations ago, which sounds almost too on the nose given what this investigation is trying to find. For a moment, the story writes itself. But legend is not lineage, and the genomic comparison placed them near the target without landing on it. Close, but still one step removed. A myth that pointed in the right direction without being the answer itself. Then came modern Tibetans from the plateau proper, the population most people would have guessed from the start. And this is where it got genuinely tight. The ancient Himalayan genomes and modern Tibetans share deep common ancestry, with gene flow between their populations continuing until roughly 9,000 years ago, before the branches finally separated for good. 9,000 years is longer than the pyramids have existed, twice over. This was the closest call in the entire comparison. Close enough that a less careful analysis might have stopped right here and called it settled. It was not settled. One population remained. The fit was not close, it was exact. Layer that population's ancient DNA against the modern genome, and the statistical distance does not just shrink, it collapses. Every marker researchers use to separate populations from one another simply stops separating. Where every other candidate left a measurable gap, however small, this one leaves none worth reporting. The comparison had been narrowing for thousands of years of eliminated candidates. Then, for the first time, it stopped narrowing entirely. The population left standing was the Sherpa, a skeleton pulled from a cliffside cave in Mustang, buried more than 1,000 years ago, is genetically almost indistinguishable from a Sherpa guide alive on Everest today. Not a cousin population, not a distant cultural relative claiming shared heritage. The direct genetic line, one population, one place, largely unbroken across more than a millennium. The title's question has an answer, and the answer is specific, but identity was never the hard part of this story. Because being descended from the people who lived in these mountains and being built to survive them turned out to be two separate facts, and they did not arrive on the same timeline. The gene most responsible for that survival is called EPAS1, and it regulates how the body responds to low oxygen, essentially telling red blood cell production when to hold back. It did not evolve inside this lineage at all. It came from Denisovans, an extinct human relative that interbred with our ancestors tens of thousands of years before anyone had ever seen a Himalayan peak. Their cousins, not their parents. A different species entirely, contributing a working piece of code that stuck around long after the species itself vanished. Here is where it gets stranger. When researchers checked the ancient Himalayan skeletons themselves for that exact gene variant, the oldest bones, the ones dated to roughly 3,150 years ago, did not carry it. Only the younger burials, dated between about 1,750 and 1,250 years old, showed the variant, which means the earliest people in this lineage were already living at extreme altitude for something like 1,000 years before they acquired the trait now treated as the defining feature of Himalayan biology. They were mountain people before they were oxygen-adapted mountain people. The order runs backward from how the story usually gets told. Separately, geneticists estimate that natural selection actually began favoring this gene around 8,900 years ago, meaning the trait was still spreading, generation by generation, long after the population had already settled these peaks and built lives there. The identity came first. The adaptation caught up centuries later, borrowed from a species that never once stood on this ground. The genome comparison is not the only place this shows up. Physiology confirms it independently, in living bodies, decades of clinical measurement. Modern Sherpas display something that looks backward at first glance. Faced with low oxygen, most human bodies respond by manufacturing more red blood cells, thickening the blood to carry whatever oxygen it can find. Sherpas do the opposite. Their hemoglobin stays comparatively low, a blunted response that looks like underperformance until you understand what it is actually doing. That pattern lines up precisely with what the Denisovan-derived EPAS1 variant is known to do at a molecular level. Two completely different fields, genetics in one lab and blood work in another, converging on the same conclusion without either one setting out to prove it. Compare that to the Andes, a different high-altitude population on a different continent entirely. Andean highlanders adapted to their mountains too, but never received this particular gene. Their solution runs the opposite direction: thicker blood, more hemoglobin, brute force where the Himalayan route uses restraint. Same problem, two different species-level toolkits, arriving at survival by separate paths. The numbers make the contrast concrete. At comparable altitude, Tibetan men average around 16.9 grams of hemoglobin per deciliter of blood. Andean men average around 18.1, less than a 2-point gap on paper. In a body starving for oxygen at 4,000 meters, that gap is the difference between two entirely different survival strategies. Alongside EPAS1, A second gene, EGLN1, shows the same signature of strong natural selection in this population, reinforcing that this is not a single lucky mutation, but a coordinated genetic toolkit. And the borrowed segment of EPAS1 itself, the actual stretch of Denisovan DNA still riding along in Himalayan genomes today, runs roughly 33,000 bases long. Compare that to a language, it would be the length of a page, embedded whole inside a shelf of encyclopedias, at a spot where evolution decided not to change a single word. You have probably felt a mild version of this yourself, on a flight, when cabin pressure shifts and your ears pop and your head goes faintly foggy for a moment. That is roughly what 6,000 feet of elevation does to an ordinary body. Sherpas stand on top of Everest, 8,849 meters, without bottled oxygen, using a gene that a different, extinct species carried into a human bloodline tens of thousands of years before either of them had ever seen snow. Every mountain that lineage has ever crossed, they crossed partly on borrowed biology. Every breath at altitude that should not work, works anyway, because of a species that is no longer here to take credit for it. The bones in that cave match the living guides on Everest today. The gene that lets both of them breathe up there never belonged to either.